We don't do it. It's fine. Yeah.
You guys ready?
Around the other side.
Tēnā koutou katoa, ngā mihi nui I tēnei rā. Welcome to our 4th Conservation Week live stream. We are at the ICMP collection today in Auckland, at our Auckland site, and we're joined by Bevan Weir, who is a lead researcher here, and he is a fungi bacterial researcher. So he's going to show us around the collection. It's a collection which contains thousands of live cultures and bacteria, pathogens as well. So it is home to one of the biggest collections internationally of all plant pathogens.
That's right.
Am I correct in saying that? Yes. So welcome, thank you so much for joining. Please type your questions in as they come. Bevan is an absolute expert and would love to answer them. So kia ora whānau, welcome.
Thanks. Yeah, so this is the National Culture Collection of New Zealand. So what is a culture? So in this way, we're talking about a culture, which is like a pure sample of a fungus or a bacteria, as you might grow it on an agar plate. So fungi and bacteria everywhere, all around us, on our skin, in the environment, growing on and in plants. But we take just a single kind of pure strain of it and store it in this collection forever. And so in this room over here are about 20,000 cultures all stored. And it's perhaps not much to look at because we can't see them because they're all preserved inside these dewars. So these containers are called stainless steel dewars, and they're filled with liquid nitrogen. And liquid nitrogen is a really, really, really cold liquid that allows us to preserve the cultures in a frozen state. So your freezer at home that you put your meat and frozen vegetables in is about minus 20 degrees. So even that freezer at home is much colder than it ever gets really anywhere in New Zealand. But liquid nitrogen is about minus 196 degrees, so almost minus 200 degrees. So it's really, really, really cold, and it allows us to preserve these cultures hopefully forever. And the advantage of doing this is that when we take them out again, they're alive. And so we can do all sorts of experiments with them. We can put them into a plant and see if they cause disease or not, or look for some beneficial compounds that they might have. So the way that we store these, and so in each of these tanks is this metal rack with plastic boxes in them. And in the box, so this is a cryovial. So this is kind of specifically designed to store frozen stuff cold, but we don't use these. We use these, which are tiny little heat-sealed segments of plastic drinking straw. So just like the drinking straw that you can buy at the supermarket, exactly the same thing. And the plastic of these is kind of amazing that I can heat this plastic up to 121 degrees at twice atmospheric pressure to sterilize it, and we can cool it down to minus 200 degrees and it's absolutely fine. And most people just use it for about a minute, when they're drinking their Coke at McDonald's or something like that. So yeah, that's why it's become kind of, people want to ban these because, the plastic is degrading, lasts forever. So that's why I've just bought about 50,000 of them.
Wow. So what kind of pathogens are in here? The likes of Cody dieback and also international ones?
Yeah, so we started this collection in about 1950, and so it's got bacteria and fungi since that time. So for the bacteria, we've got almost every single bacterial plant pathogen that's ever been described in the world.
All in this room?
Yeah, all safely kept in this room, which is why you guys are not going in there. And that's really useful. And so one example where it became really, really useful was in the PSA disease of kiwi fruit a few years ago in 2010. And so at that time, we didn't know if the PSA was what it was or if it was a new disease or not. But in here, so a Japanese guy, Takikawa, in 1980, when he first found this kiwi fruit pathogen in Japan, he sent us a culture. And we put it in here and kept it safe for all those years, 40 years, until, this disease struck. And then we work a lot with MPI, and they were able to come in, we can take that culture out and test it and compare it against what we had in New Zealand. So at the same time, we also had a lot of cultures that were isolated from kiwi fruit over the past decades as well. And so we could look back through that history of time and find out if PSA was a new incursion into New Zealand or not, or if it had been existing for a while and just some kind of circumstances at the time. And
to give people an idea of how far spread the collection contains material from all over the world, these little dots represent how many collections are from a particular place?
That's right, so each one of these dots is one culture. So you can see we've got stuff all around New Zealand, but it really is an international collection with fungi and bacteria from all over the world. So while we're here, for each of the cultures in there, we've got a web page that has all the information about them, so kind of what it looks like, who figured out what it was called, where it was collected, and some additional information, maybe if there's gene sequence data. So, for example, this is one of the cultures that's had a DNA sequence done. In some cases, we do whole genomes as well. So these cultures are used by many people all around the world. So it's a really active collection. So we store about 1,000 cultures every year from both our own research and other people wanting to deposit their culture in here to keep it safe forever. And at the same time, we send out about 600 cultures around New Zealand and the world for people who are using these cultures for their research.
Cool. Now we're really lucky, and Bevan's going to actually go inside and give a demonstration of what these racks look like, because they don't look as clean, they're very clean, but they look very icy inside the liquid nitrogen. So let's take a little look.
Cool. So we're in security, so I have to kind of swipe myself in and get ready. So liquid nitrogen, being minus 200 degrees, is kind of a dangerous substance, so you don't want to get it on you, so I have to get all my protective gear on. And liquid nitrogen is the same thing that the doctor uses in the surgery if you need to get any warts frozen off, so you really, really don't want to get it onto your skin. So putting on my lab coat and my big thick gloves, and inside these dewars, if we lift it up, we can see all the liquid nitrogen making the air and the moisture in the air frozen. Inside we can see these racks. So it's the same white boxes with those straws with the cultures kept in them, and completely frozen and safe. And you can see we keep the bottom few boxes free, so most of it is in the gas phase of liquid nitrogen. Just pop that back in again. It's kind of a knack to getting it in. There's a lot of safety that we have in this room. So there's both the kind of danger of getting nitrogen splashed on you, but liquid nitrogen also has a real problem for breathing. So the air that we breathe right now is about 70% nitrogen, but 1 litre of liquid nitrogen, when it goes into a gas, expands 700 times. And so that means that this is a problem. If we get too much nitrogen building up in this room, it can displace the oxygen. So we've got safety of having some really, really high airflow in here that replaces the air every minute. And we've got various sensors that detect the oxygen level. So I'm just going to walk through the lab and show you some other stuff. So your freezer at home is about minus 20 degrees and liquid nitrogen is about minus 200 degrees. And these freezers are somewhere in between, so these are about minus 80 degrees. And so these are also a really good way to keep things frozen and preserved for a reasonable amount of time, several decades, but not quite as long as liquid nitrogen. As a backup, each of these freezers has liquid carbon dioxide that can get squirted in if we ever have a power failure. And I'll just let you guys into the lab to have a look around. Come on in. So this is a microbiology lab. So in this lab we take those cultures from whatever kind of environment there might be and we grow them up and pure. And this lab's quite special because it's a containment facility. So we can take in cultures from all around the world, nasty pathogens, and work in here really, really safely. So we've got sterile air called laminar flow cabinets which blow sterile air down on so the plates don't get contaminated at all. We've got various freezers and incubators so we can run our experiments for growing fungi at any kind of temperature or humidity conditions that we need. And in here we have a growth room. So the human body is, with about 36 degrees, and so human pathogenic bacteria grow about that. But most of the environmental fungi that we have in New Zealand, that's way, way too hot. And that's why our body temperature is high, to kind of kill fungi that don't normally grow at that temperature. So in here we keep it about 18 degrees. And the lighting conditions are really important as well. So we have these lights with a kind of a near UV and white light, and they're on a 12-hour timer. So they just replicate the normal sunrise and sunset. And that lets some fungi be able to produce spores and to be able to fruit. So this white stuff is the normal kind of the thread growth or the mycelium of the fungus. And then under the right conditions, so hopefully under this light conditions, we get it to make these little fruiting bodies with all the spores on top, which can then in nature that would kind of float away, land on something else and be able to produce a new fungus.
So you can take cultures out of the liquid nitrogen and grow them, put them in an agar plate and grow them in here.
That's right, yeah. So when we store them in liquid nitrogen, we freeze them down quite slowly and carefully. And this is just a thought experiment, please don't do this at home, but if you think about if you caught a mice or a rat or something like that and you put it into your freezer at home and you take it out again, it's probably not going to come back to life, right? And the reason for that is that the water in the cells of things, when you freeze it, the water expands and bursts open the cells, killing them. And so with fungi and bacteria, we have that same problem. So what we do is we mix in a cryoprotectant, we call it, called glycerol, and that means when the water in the cells freezes, it doesn't expand. So we can store it in that liquid nitrogen forever. When we want to take it back out again, we need to heat it up really fast in some warm water, and then we can squeeze it onto a plate and take it in here and to grow it up to do our experiments or send it off to whoever's ordered that culture for their own research.
So you've taken some and grown them for us. So let's have a look at what you've got in the plates.
Yeah.
And as you were just saying, you can send these overseas to researchers as well?
Yes, yeah. So we send them overseas all the time, and so people might need it as a control. So they want to, maybe they think they have a particular plant pathogen, but they're not quite sure, and so they can order one of our ones that's a known type of a pathogen, so they can compare with it. or yeah, maybe they want to do some kind of experiment with the properties of it. Maybe it produces antibiotics or something like that. And so I've got a project with Dr. Susie Wiles at Auckland Med School, where we're going through the culture collection at the moment and looking at some of our native fungi and seeing if they produce new antibiotics that we're able to use in medicine.
It's really interesting because obviously penicillium won't be the only bacterial, well, culture that we can use.
Yeah, so unfortunately there's been some overuse of antibiotics in agriculture and horticulture even. And so there are, when you do that, some cultures can become resistant to antibiotics. And so we have to keep kind of finding new ones. And hopefully if we find a new one, we'll be a little bit more careful about how we use it.
Okay. Let's have a look at what you've got over here.
Yeah. So again, we've got a few different kinds of cultures. And so this one is quite similar to what we saw before. So funnily enough, this is actually penicillium. So they don't all form this kind of crazy fruiting structure, but this one's quite cool, and it does. And so again, you've got these stalky things, and that blue stuff on the top there is the fungal spores, which in the environment would kind of float off and land on something else and be able to grow again.
And so penicillin is something that people would be quite familiar with antibiotics, but they probably wouldn't realize that it's an extract from this.
Yeah, that's right. So Alexander Fleming in the UK many years ago was the first person to discover these antibiotics. And so, and it's funny, and we've had it in the lab as well, and it was just kind of an accident. So he was growing up some bacteria for an experiment, and he got this contamination of penicillium on his plate, and it started killing his bacteria. And I thought, oh, that's interesting. And so from that, he was able to extract the kind of active compound, which we called penicillin, out, and commercialize it into an antibiotic and save so many people's lives. So that's one example of what fungus looks like growing pure on a plate. This is another one, and so it's got this kind of typical fluffiness on it, which is the fungal mycelium, we call it, or just the kind of fungal threads that are growing. And this one also produces a interesting, really red compound, and this is the fungus Epicoccum. And this red compound is also antifungal, so this will be produced to kill other fungi. And they don't do it to, make medicine for us. These fungi are doing it because they want to exist in the environment and fight off other fungi so they can grow. We're just kind of exploiting that property. And
so what are some of the interesting research projects that are happening at the moment? You've mentioned the new antibiotics, but obviously Cody dieback is very topical, and as you said, this is the largest collection that we have of Cody dieback.
Yeah, so there's a lot of people around New Zealand working on Cody dieback, but the research that I did was describing it as a new species. And so it was first kind of found in about 1970 in Great Barrier Island, but we didn't realize it was a new species until quite recently. And so I did a lot of work around looking at the genetics of it and the morphology and figuring out that it was a new species, which I gave the very complicated Latin name of Phytophthora agathidicida, which in Latin just means the cowrie killer. Some other projects that we have on at the moment, I'm looking at the microbes that are in kombucha. And so kombucha is a kind of semi-popular drink, and it's got a mixture of yeasts and bacteria in it. And so this is one of the yeasts that will be in it. So this is the same yeast that you can brew beer with or bake bread. And so this yeast feeds on the sugar in kombucha and turns it into alcohol. And then the bacteria in the kombucha eat up that alcohol and turn it into vinegar, which makes that kind of sour taste of the kombucha. So yeah, I'm just really interested in what, because this is a commercial bottle, But there are so many home brewers around New Zealand that are making their own kombucha. I'm just really interested to see what people have got kind of growing in their backyard, and it's coming from the New Zealand natural environment, and we've got our own unique kombuchas.
You've also brought in some blue cheese.
Yeah, so bacteria and fungi are everywhere, and we use them all the time. And so blue cheese is something that many people will know of. And so the blue cheese has got, again, the same fungus, penicillium, in it. And if we break it open, we can see the blueness of the cheese is actually the fungal spores. And so it's inoculated, and so we've got fungal spores in there, and throughout the whole cheesy part of it are the mycelium. And same kind of deal with like camembert cheese as well. It starts digesting the cheese a little bit, breaking down and making it nice and soft, and it gives this kind of tart flavour. So if we look at some of this, if we make a slide and put it under the microscope, and I've got just a little microscope here, so we've squished it onto a slide and put it under there, and you can see that this part is the cheesy part, so it's got all the cheese proteins and fats, and in the middle here we can see the fungal spores. But this is absolutely safe to eat, of course. People eat blue cheese all the time. And that's kind of my point, so we eat microbes all the time in kombucha, beer, bread, blue cheese. Yogurt has got different kinds of bacteria in as well. And so some can be bad, so some can be pathogens, but some can also be really good and useful for us. This fungus is called trichoderma, and this is a really, really useful fungus. It's used to protect plants from other fungi. So if you introduce this into a plant, it'll colonize the plant very safely, won't hurt the plant at all, but produce compounds that will protect it against plant pathogens.
So to help look after the collection, obviously when researchers come and they want to take some cultures and they want to experiment on their own for their own research, you'd have to continue to grow the collection while you keep it here and house it. If everything got used, it would be all used up. So how do you preserve it?
Yeah, so when we store it down, we make about 10 of those little drinking straw things and split it into 2 different containers for safety. And so when someone wants it, they can just pick up one of the little straws and we'll culture it for them. And when we get to the last one, we can just take that plate and restore it back again. So we're always kind of cycling it and keeping it alive. And of course, we're adding in our own new cultures, and people send us our own stuff as well. So we kind of grow the collection like that.
So how old are some of those samples?
So the New Zealand ones, the oldest ones, are about 1950s. But for example, we do have that penicillin producing strain from Alexander Fleming, so that's much older. It was a sort of production strain, say 1940s or so. We don't have really, really old things because in the past people didn't have the technology to keep cultures alive for that long. And so when this collection was going in the 1960s, we didn't have that kind of freezing technology. So we had to keep culturing it year after year. So take a bit off a plate, then put it onto a new fresh agar plate all the time. And that was, really, really kind of labour intensive.
And so you have a really cool demonstration to show us about liquid nitrogen. Shall we have a little look?
Yeah.
What happens?
Why not? So, I was talking about why we have to go this kind of really special procedure to store things down and using this cryoprotectant glycerol. And it's because, so this is a pot of liquid nitrogen, so you can see it in there. And so liquid nitrogen, really, really, really cold, about minus 200 degrees. And so this is what would happen if you put your finger or something like that in it. So right now this liquid nitrogen is cooling down the flour, and we can kind of see it kind of bubbling away in there. And it's becoming frozen. You can even see the colour change in the leaves there as well.
It's happening really quickly.
Yeah, so it's opened up. And so that's completely frozen now. And let's see what happens. You can see it's been completely frozen. It just kind of turns into a powder.
Very cool.
And so that's what would happen if we didn't use that cryoprotectant to protect those cells of the fungi and bacteria as well. They would just become all brittle and just die and crumble up when we took them out. So doing that is, yeah, really, really important.
And so because this is a live collection, you'd use a lot of liquid nitrogen.
We do, yeah. And so because liquid nitrogen is so cold, and you can see it right now, all this vapor coming off, we fill up the tanks, and just the warmth in the room makes it boil off. And so that means we have to keep topping up the tanks. And so every 2 weeks we go through about 400 litres of liquid nitrogen. Yeah.
And so the collection is also digital, as we spoke about earlier. And the digitisation process happens here as well, where you capture photographs?
Yes, so I guess there's different parts to that digitisation. So we've got collecting the information about where and when it was collected, and we do all that in a form, submission form, when people send us the cultures. We also, there's another lab downstairs where we can sequence genes or genomes, and also really important is to see what they look like, because different fungi can have kind of different colours, and that might mean they produce different kind of compounds. So for example, this Epicoccum one with the red colour, if we wanted to capture this in an image, we've got it's a little photography station where we can, set it up and take a photo and then put that on the website so people can see some kind of morphology. And we can also do microscope photos as well, to take photos of the spores or anything else that's needed.
And so this is such a valuable research, or this is a collection really valuable for research. How would you see, if we didn't have something like this, a resource that wasn't preserved for use in the future, in the past, going forward? Like, what would it look like for science?
It would be really problematic, and I kind of know what it's like, because Australia, a near neighbour, doesn't have any national culture collection, and so we get a lot of work with them because they really need somewhere close to store their precious cultures once they've done research, or to access for their own research as well. So if we didn't have this collection, it would really, really inhibit the ability of scientists to do research on fungi and bacteria in New Zealand. And so it's really important for biosecurity reasons. So we've talked about the PSA on kiwi fruit and Phytophthora on causing kauri dieback. And so that would, if we didn't have the culture collection, that would inhibit our ability to respond to these plant pathogen incursions. But also on the conservation side as well, there's a lot of native fungi, and some of them are rare and endangered, and so we can keep those alive, hopefully forever, in a safe place if they're ever needed.
Okay, so you can bring them out and grow them and
Yeah.
use them. Yeah, and so this is obviously a very secure facility that we have, because the other collections, are housed in the same building, which is
Yeah.
quite interesting, given there's such strict parameters around contamination. So for a facility like this, obviously people can't just come and visit.
No. So we've got, even to come into this lab and do work, you need to go through kind of an induction process. You have to get signed in, there's swipe card entry and all that kind of thing. And then we've got another layer of security, again, with dealing with cultures. And There's regulation about the cultures as well. So anything coming in from overseas is kind of a quarantine culture, and then MPI puts on other layers of regulation on top as well. So, for example, really nasty things like Kauri dieback is considered an unwanted organism, and so you have to have a special permit to even be able to work with that as well.
And then people would, at the moment, use that culture, and they would test it against others to see what could be resistant. Is that the kind of work that's happening?
That's right, yeah. So there's a scientist, Monica Girth, in Otago University, who is using some of our Kauri dieback cultures and testing plant extracts from native plants to see if they're able to kill the Kauri dieback disease or stop it from affecting kauri. Yeah.
Let's have a little look at some of the other things, other cultures that you've got
Okay.
over here. They're really cool.
Yeah, so there's kind of a wider range of colours and shapes, and so this is kind of a cool one because I think it kind of almost looks like a flower type shape when it's growing on the plate. And some of the phytophoras that cause the disease like Kauri dieback also have this kind of morphology and shape when they're growing as well.
How long does it take when you take a culture out of the liquid nitrogen to get it to grow this much?
Yeah, it's variable. So these are probably all done at the same time, so we can see the kind of difference. So just in the few days, you can see one's filled up the whole plate and one's only grown, sort of a centimetre or so. So normally somewhere between 1 to 2 weeks to grow. So, and this is kind of an interesting one, and so this is a really, really bright pinky red yeast that's quite common in the environment, but you just wouldn't know it because when it's growing on a plant, it's kind of microscopic and you'd never see it.
Okay, cool. Well, thank you so much, Bevan. Thank you for giving us an excellent tour of the ICMP collection. Ngā mihi nui koutou, thank you for tuning in. If anyone has any questions that they haven't asked during this session, please type them and we'll get back to you. And for more information, you can also go onto our website and go onto iNaturalist to see where these are stored online. But thank you so much for tuning in, and thank you again, Bevan, for giving us such a great tour of the ICMP collection.
Cheers.
Ka kite.